Hi, good afternoon, everyone. Thank you so much for joining Jet Foundation's Community Workshop Series. We are going to just wait a few seconds here and allow some more attendees to begin, to join us. We'll start up in a few. All right, hello everyone again. Good afternoon. Thank you again for joining our webinar series. My name is Danielle Edwards. I'm Director of Community Engagement at Jet Foundation. I've been a part of this organization now for almost 5 years. This year, our community webinar series will be on helping our Duchenne community be better prepared. While I wish we could be doing this in person, our traditional family workshops, I'm so glad we're able to provide you with these virtual webinars. Today, I'm so excited to have Dyne Therapeutics with us, who'll be presenting on an overview of its FORCE platform and Duchenne drug development program to our Jet community. Please make sure to drop any questions or comments you may have for them in the chat below so that they can answer after the presentation. With that being said, I hope everyone enjoys the webinar today, and I'll pass it over to you, Mo, to begin.
Okay, good afternoon, everyone. First, I want to thank the Jet Foundation for the opportunity to share Dyne's vision with you today. We always feel privileged to be working in an industry where we get these opportunities to speak directly with patient communities. At Dyne, our mission is to bring life-transforming therapies to patients with serious muscle diseases, and at the forefront of that is our goal to transform the treatment paradigm for Duchenne muscular dystrophy, or DMD, which, as we all know, has profound unmet needs in the current available treatments. This is the least favorite slide in my presentation, but I have to show it to you to keep some lawyers happy. I won't read the whole thing, but in a nutshell, it says that we will be making forward-looking statements that involve risks and uncertainties. And for more information, please refer to our SEC filing. At Dyne, it all starts with our mission. We are laser focused on delivering life-transforming therapies to patients with serious muscle diseases, with DMD being a priority. Our mission is core to who we are and what we do each and every day. A commitment to understanding the community of people living with muscle diseases, and DMD in particular, ensuring that their perspectives and experiences are included in our drug development programs is central to our work. We created a multifaceted program involving community webinars, conference participation, fireside chats, and other outreach to raise visibility and gather input to advance our drug development efforts. Examples include hosting the World Duchenne Awareness Day fireside chat, Dyne Rare Disease Day fireside chat this year, and many other outreach efforts to the DMD community. Your voice is important to us. Over the past couple of years, we have been validating our platform and technology, including our ability to target delivery of potential therapeutics to muscle, and have generated exciting preclinical data across multiple indications and disease models. And our focus today is on sharing our data on our DMD program. We are building Dyne as the leading muscle disease company to deliver for families living with DMD. Shown here are 4 core strengths to help us deliver on that. 1, we intentionally designed our FORCE platform to overcome the current limitations of muscle tissue delivery with modern oligonucleotide or PMO therapies. The goal for our DMD program is to develop therapies for multiple skip-amenable DMD mutations that significantly increases dystrophin production and enable less frequent dosing to potentially stop or reverse disease progression. Second, precision medicine targeting the genetic basis of DMD that we have already proven preclinically to produce significant increase in exon skipping and dystrophin production and meaningful improvement in function in disease models. And I'll show you some of that data in this presentation. The third core strength is our clinical plans. They're being built to ensure that we incorporate patient burden and family needs regarding clinical study design. We are developing therapies for multiple exons, rapidly advancing our lead exon 51 program, followed by exon 53, 45, and 44 to reach additional patients. And we are completing studies and working with regulatory agencies to prepare for our clinical trials. 4, we have exceptional in-house and advisory expertise with deep knowledge of DMD and novel therapeutics and a world-class scientific advisory board. For example, Francisco Mantoni on our SAB is a world-leading physician and a clinical trials leader involved in multiple DMD trials. We also have Dr. Lou Kunkel, who is the scientist that discovered that defects in dystrophin are the cause for DMD. This is a schematic that depicts our FORCE platform, which is the technology we use to deliver potential therapies to muscle. Our FORCE platform consists of a specific delivery mechanism called a Fab, or an antigen binding fragment, that we have engineered to bind to a receptor called TFR1 that is found across the surface of muscle cells. If you look at the Y-shaped structure on the left, that is how a full antibody looks like. Our FORCE platform uses the top piece, the Fab, which drives binding to the TFR1 receptor on muscle cell walls. We believe Fabs have several advantages over full antibodies or mAbs, including smaller size, which helps with distribution to muscle tissue. This approach is intended to take advantage of specific muscle biology, allowing targeted delivery of the PMO and potentially avoiding off-target effects in the liver, kidney, and other areas. The Fab acts as a kind of key that unlocks the TFR1 receptor and allows our therapeutic PMO to enter skeletal, cardiac, and smooth muscle to target the genetic cause of diseases. We link our Fab to the potential therapy, in this case PMO, with the linker depicted as the yellow line. The linker molecule connects the Fab to the PMO depicted as a ribbon in the schematic, which in turn drives exon skipping and is designed to target the genetic basis of DMD. Exon skipping PMOs have been shown to produce very modest amounts of dystrophin and are approved in the US. However, historically, the challenge, as stated earlier, has been the inability to get enough of these PMOs into the muscle cells to do a good job in skipping and driving dystrophin expression. These are what's called naked PMOs, meaning they do not have a specific delivery mechanism that helps to direct them to the appropriate tissue. Our FORCE platform is designed to solve the delivery issue and deliver therapeutic payloads into muscle cells by attaching the potential therapy, PMO, to our Fab, which enables targeted delivery to muscle. This is the advantage of FORCE that will carry the PMO to muscle cells compared to naked oligos, as we will see in the next video.
At Dyne, we are focused on advancing life-transforming therapies for people living with serious muscle diseases. We are utilizing our FORCE platform to develop modern oligonucleotide therapies that are designed to overcome limitations in delivery to muscle tissue. Our therapeutic candidates consist of 3 components: an antigen-binding fragment antibody, or Fab, a clinically validated linker, and an oligonucleotide payload. The Fab is conjugated or connected to the therapeutic payload using a linker. Our proprietary Fabs are engineered to bind to the transferrin 1 receptor, or TFR1, which is highly expressed on the surface of muscle cells. The Fab engages with the TFR1 to facilitate the delivery of the payload into the muscle, where it aims to target the genetic basis of disease. Dyne is advancing therapeutic candidates for myotonic dystrophy type 1, Duchenne muscular dystrophy, and facioscapulohumeral muscular dystrophy. The modularity of the FORCE platform enables the selection of different types of payloads to match the genetic basis of the disease, including antisense oligonucleotides, or ASOs, in DM1, phosphorodiamidate morpholino oligomers, or PMOs, to promote nuclear exon skipping in DMD, and small interfering RNA for targets in the cytoplasm. With the FORCE platform, Dyne is aiming to deliver therapies with the potential to transform the futures of people living with serious muscle diseases. At Dyne,
There you go. We believe our FORCE platform delivers significant advantages with the ultimate goal to stop or reverse progression of the disease. It drives targeted delivery to muscle through conjugation to the Fab that targets TFR1, a receptor that, as mentioned, is highly expressed on muscle cells. This leads to enhanced delivery to skeletal, cardiac, and smooth muscle. It targets the genetic basis of the disease, and in this case of DMD, the goal is to induce exon skipping of the mutation that is the cause of the disease to induce the expression of the dystrophin protein. It is redosable therapy with potential for individualized patient titration. It also has enhanced tolerability due to targeting delivery to muscle and potentially avoiding off-target effects in the liver, kidney, and other areas. We have exciting preclinical data that shows prolonged duration after a single dose, supporting a potential once-monthly or less frequent dosing. And finally, the modularity of the platform, meaning using the same Fab and linker and just changing the oligo depending on the disease or mutation, gives our platform significant efficiencies in reducing development and manufacturing costs. The goal for our DMD program outlined on this slide is to build a best-in-class exon skipping franchise that significantly increases dystrophin production and enables less frequent dosing to potentially stop or reverse disease progression. Our FORCE platform is designed to overcome the significant limitations of current therapies, and we've generated very exciting preclinical data, starting with our lead Exon 51 program, where exists high unmet need despite approved therapies. This data from a study with a lead conjugate in DMD patient cell line that is amenable to exon 51 skipping. FORCE achieved enhanced exon skipping in blue compared to their naked PMO, which is represented in red. The naked PMO achieved about 20% exon skipping compared to about 50% for the Dyne FORCE molecule. We build on our work in patient myotubes to evaluate the FORCE platform in the MDX mouse as a well-validated model of DMD that you'll see on the next slide. This slide summarizes the power of our FORCE platform in delivering to muscle and driving significant exon skipping versus what has been published on naked or non-targeted PMOs. As shown in this slide, FORCE achieved more effective exon skipping than other approaches in this DMD mouse model, leading to higher delivery of these therapeutic PMOs to muscles and much better exon skipping in 3 key muscle types in DMD. This mouse model has a mutation in exon 23. With this mutation, there's no dystrophin, and it can't run as well as wild-type animals, and it has high creatine kinase in the serum due to muscle damage and has a shorter lifespan than a wild-type mouse. This model enables comparison of our FORCE platform in blue squares with published data from a naked PMO in the red triangle and a stereopure antisense oligo, or ASO, in the orange triangle. We observed significant exon skipping in the quad, up to 66%, after a single dose of FORCE at 10, 20, and 30 mg/kg, 2 weeks after dosing. Not only do we see enhanced exon skipping in the quad, but you also see enhanced skipping in the diaphragm and heart, which are really important muscles for DMD, and where other modalities had trouble achieving skipping with single doses. As you can see for the diaphragm and heart, You barely see any skipping with other modalities, the naked ASOs or PMOs versus force, where we see in the diaphragm up to 44 or 45% skipping, and in the heart up to 35% skipping after a single dose. This increase in exon skipping resulted in dose-dependent increase of dystrophin expression that you'll see in the next slide. Exon skipping is the first step. So we wanted to look at what happens after we enhance delivery and skipping. What happens to making dystrophin, the actual missing protein? Once the PMO makes the cell skip the mutation, we should start making dystrophin protein so that the protein can attach to the surface of the cell and do its job and provide support to the muscle cells. This is exactly what we see in this slide. On the left is Western blot, and on the right is a histogram which quantifies dystrophin levels. Our highest dose of force resulted in 9 to 10% dystrophin with no demonstrated dose-limiting toxicities, while naked PMO produced around 1% at a higher dose of 40 mg per kg versus the 30 mg per kg in our highest dose. This is a single dose experiment, and given that DMD is a chronic disease, we believe that our redosable, titratable therapy, we can leverage TfR1 mediated cell entry to redose and continue to build up dystrophin levels. These studies are ongoing, and we will be happy to come back and share the data with you as it becomes available. Once the cells make dystrophin, we need to make sure that the newly made dystrophin locates to the surface of muscle cells where it can do its function. And this is what we look at as shown in this slide. Immunohistochemistry studies conducted 4 weeks after that single dose study we showed earlier demonstrated that we restored dystrophin expression on the cell surface or sarcolemma, which is important to restore muscle cell integrity and restore function as mentioned earlier. You can see that in wild-type animals, there's a nice staining on the surface of the muscle cells for dystrophin. This is on the far left. This goes away in the MDX mouse model that does not produce dystrophin, which is the panel right next to it. On the far right panel, this panel demonstrates that force increased dystrophin positive fibers, and dystrophin is located to the sarcolemma, which would restore muscle cell integrity. Once you enhance muscle cell integrity, we should decrease the amount of creatine kinase, or CK, that leaks out of the muscle into the serum when the muscle cell is damaged, which you'll see in the next slide. So creatine kinase, or CK for short, is a clinical biomarker of muscle damage. Here we show that a single dose of force demonstrated significant decreases in serum CK at 2 weeks and 4 weeks post-dose compared to a naked PMO. If you compare the blue force conjugate with the red, which is naked PMO, this is evidence that we are providing benefit to these muscles and repairing muscle membranes. Subsequently, we evaluate how these data translate into the functional benefits, which is the ultimate measure of the ability of any therapeutic to potentially stop or reverse disease progression, which will be shown on the next slide. On this slide, we show the functional test that we conducted that shows that the dystrophin being made is actually working on the cell surface to provide the support and function needed. We conducted blinded studies to evaluate functional benefit in the same MDX model. On the left is a running wheel test. The y-axis represents distance traveled, and the x-axis is time of the day. Between the two vertical dashed lines is the dark period, when mice, which are nocturnal, are active at night, run on the wheel. The gray circles are wild-type mice, the black squares are MDX model, and you see the difference in activity. The MDX mice fatigue easily and don't move as much over the whole 12-hour period. Force, in blue triangles, restored function in MDX mice comparable to wild-type mice. This is not at a single time point, as this activity is over 12-hour period. We believe this supports a benefit to skeletal and cardiac muscles, since we are enabling these MDX mice to increase activity over a 12-hour period. The naked PMO in the red triangles did not seem to improve function in this study at all at a single dose. On the right, you can see the results of the hind limb fatigue challenge, where mice were required to stand for 10 minutes on their hind limbs, and then we measured loss of range of motion. The gray bar is the wild type mice, which loses about 20% range of motion. MDX is the black bar, which loses 70%, with similar data for the animals injected with naked PMO. And as you can see, the Dyne force conjugate treated MDX mice in blue regained the range of motion that is now almost equivalent to the wild type range of motion. These are very exciting data. We are focused on building a global DMD franchise, rapidly advancing Exon 51 program, followed by 53, 44, and 45 to reach additional patients. We have an opportunity to target up to 80% of DMD population that's amenable to exon skipping therapies, and hope to serve as many affected families living with DMD as possible. Just to summarize our progress and data, we are excited by the compelling data in the DMD program that are driving excitement in the field. Preclinically, we have achieved effective exon skipping in muscle, dose-dependently increased dystrophin expression, and reduced serum CK, a clinical biomarker of muscle damage. And we have demonstrated functional benefits in multiple standardized assessments. We have also increased exon skipping versus unconjugated PMO in human DMD myotubes. We are looking forward to finishing our preclinical studies and finalizing our clinical plans for DMD exon 51, which is one of 3 INDs planned in the time window between end of this year and next year. We also have an opportunity to advance our DMD programs for exon 53, 45, and 44 to reach additional patients. As mentioned earlier, our ongoing outreach and discussions with the DMD community have been critical to ensuring that we understand the disease burden as it is experienced by boys and men and their families, the complications and needs of families participating in trials, and all of the elements that become part of the DMD experience. We've captured a few of the comments you have shared with us here. I want to thank all of you for sharing your personal stories. You don't know how much this means to us. Your feedback makes us better scientists, better drug developers, and better partners, and we feel a tremendous sense of urgency to deliver for individuals impacted by DMD. Finally, I can't thank you enough for giving us the opportunity to speak to you. Dyne's sincere commitment to patients will always guide our path as we build our programs and the company. When Ravi, Beth, Diana, and their families speak to us and share their stories and perspective, you don't know how much this means to us and how much we appreciate their courage and fortitude. Thank you again for the opportunity to share Dyne's compelling story with you today, and we'll be taking questions.
Thank you, Mo, for that presentation. As Mo was just saying, right now we'll be taking questions from the audience, so please feel free to drop any that you may have for Dyne in the chat section below. Give it a second. I have one question. Do you guys have any plans to do anything other than exon skipping?
For now, our plans is to do exon skipping. This is as mentioned earlier. This is a drug-like conjugate where it's titratable, redosable, and that would be our focus within Dyne. And exon skipping, as we all know, has proven, it's a proven mechanism. The challenge is getting enough of these PMOs into muscle, which we think we have solved within Dyne with the FORCE platform, and we believe it's going to work. As long as you get more of these PMOs into the muscle, it will work. It will cause the skipping, increase dystrophin expression more than existing technologies, and eventually, as we've seen in the preclinical data, lead to the location of the dystrophin to the surface and improve function.
Thank you. Another question I have is, will you move to microdystrophin?
The short answer is no. And the reason we're saying that is we believe that a skipped molecule, dystrophin, has more of the protein and would provide more benefit for the muscles, for the cells, and eventually functions.
Okay. Do you have plans to do other exons other than the hotspots?
We do. This is something that we're, as mentioned, the lead program is 51, and it will be followed pretty quickly with 45 and 53 and 44. But we have others. Our technology allows us to address all the exon amenable patients out there, and we're working on doing that and developing these therapies.
Do you have plans to enroll non-ambulatory patients in your study?
We are exploring all of our options regarding we're in the middle of designing our clinical trials with feedback from KOLs, investigators, as well as patient communities. And we're exploring both ambulatory and non-ambulatory. And we will update you as we finalize our plan, with outreach to you guys, to the community, to the physicians, as well as on our social media channels.
Great. And when can families expect you guys to begin enrolling for your trial?
So as we've discussed and we've disclosed, we have 3 INDs for the 3 main diseases we're working on between the end of this year and the end of next year. But I can tell you that DMD is on the earlier side of this. And as we progress more and we file our INDs, we'll be in touch with the community, as well as on formal channels like clinicaltrials.gov to update you on where we are.
Okay. Do you anticipate an age cutoff for your study?
This is part of the evaluations that we're doing now on the patient population we'll be targeting. That would include considerations for age, function, etc. And we'll update you as we finalize these plans.
Great. Those were just some of the questions we received. Again, if anyone has any other questions for Mo and Dyne Therapeutics, please feel free to drop it in the chat section.
Danielle, I do see a few questions. Are you able to see those?
Oh, perfect. Yep. All right. Here we go. Now we have some more coming in. How large of a payload can your FORCE platform deliver? Could it be repurposed to deliver dystrophin RNA or an in-frame subset of dystrophin RNA?
It's a really interesting question. I've actually haven't been asked that question before. We haven't tested that yet. Theoretically, possibly, but that's a really interesting. That's an interesting question. We'll definitely think about that. We haven't tested the limits yet. What we know is it can deliver oligos, which can be 30 or 40 or nucleotides, etc. But we haven't looked at the messenger RNA. Dystrophin is a pretty big protein, and the RNA is pretty big, but we haven't tested it yet. But it's something. It gives us something to think about. It's a good question.
Great. Another one, again, what is your timeline for other deletions and duplications such as point mutations? Sorry.
Yeah, I mean, what we've disclosed is we're going to initiate our IND at the earlier of that window, end of this year to end of next year for the 51. The other exons that I mentioned today are right behind it. We haven't disclosed when yet, and we'll be working on the other exons as well. So stay tuned. We'll be happy to update the community as soon as we get data that tells us that we can push forward for with the other exons.
Great. And then we just got a few thank yous for your presentation, but I think that's it right now for all the questions from the community. If anyone else has any other questions, feel free to drop it in the chat, but right now it seems like that's it. Mo, thank you so much for your presentation. It was very informative, and I hope a lot of families got a lot out of it today. If you have any other questions or would like to be connected with Dyne Therapeutics, please, respond back to our emails. We are happy to connect you and point you in the right direction. We appreciate all of you joining us today, and a huge thank you to Dyne Therapeutics for being in the space and your commitment to the Duchenne community. With that being said, we'll let everybody go a little bit earlier today. Thank you all so much again, and I look forward to seeing you on another community webinar very soon.
Thank you very much. It was a pleasure, and hope to see you again as we progress and update you on the progress.
Absolutely. Thanks so much, everyone. Again, we'll be ending this then now. Have a great day.
Thank you.